Hello, good afternoon, and welcome to the Department of Biochemistry at the University of Cambridge in the UK. I'm Dr. Rhys Grant, and I am the Science Communications Specialist here in the Biochemistry Department, shared also with the Cell and Molecular Biology programme at the Cancer Research UK Cambridge Centre at Cambridge University. So I'm extremely pleased to welcome you this afternoon on this beautiful Tuesday to our department for this live talk as part of the 2021 Cambridge Festival, which aims to bring the research of the University of Cambridge directly to you in your living rooms, or let's be honest, more honestly, in your gardens, given how nice the weather is. So for this talk, I am joined by Dr. Tony Jackson, who is a senior lecturer in biochemistry at the University of Cambridge. And today, Tony's going to be speaking to us about how molecular signaling channels work and how their failure may underlie many diseases, such as heart disease and cancer. I'm also joined, as always, by Latika Sugamba, who is part of our department's secretariat team. And Latika is going to be helping me out this afternoon behind the scenes with running this event. Before we get on to any of our awesome science though, I'd first like to ask you all to please click the subscribe button down below to follow our YouTube channel, as we have loads more talks coming up this week for the Cambridge Festival, and we're also planning on bringing you more live content from our department in the near future. I'd also encourage you all to go and check out our other social media channels after today's talk, obviously, on Twitter, Facebook, Instagram, LinkedIn, and so on, using our handle, which is @CamBiochem. So that's CAMBIOCHEM, because we use these channels to bring you all of the news, the research, the teaching, events, outreach, and information about everything really that goes on in our department. I'd also just like to mention that after Tony's talk today, Tony, Latika, and myself will be having a live question and answer session. So if you have any questions on Tony's talk, or if you'd just like to ask us something more general, maybe about what life is like in our department, if you could please post your question in the live chat on YouTube, and then the three of us will stay around for 15 to 20 minutes after Tony's talk to answer as many of your questions as we can get through. You don't need to worry if you're currently watching this talk while sunbathing in your garden or anything like that, because if you choose to participate in the Q&A session, you won't be seen or heard. It will just be me reading your questions out anonymously on your behalf. So, as I mentioned, this afternoon's talk is going to be given by Dr. Tony Jackson, who is a senior lecturer in biochemistry at the University of Cambridge. But Tony didn't begin his career in our department. He actually began it in another Cambridge department, the Department of Clinical Biochemistry. And this is where Tony completed his PhD. He then moved to the Department of Cell Biology at Stanford University in California for his postdoctoral research. Tony is now a senior lecturer in our department, and his current work focuses on voltage-gated sodium channels, and in particular, the channels that are specialized for pain perception and those that are selectively expressed in the heart. In addition to Tony's research, he's also a teaching fellow at Sydney Sussex College in Cambridge. So with that, I'd like to hand over to Tony for this afternoon's talk. So thank you very much, Tony.
Okay. Thanks, Rhys. And I'm now going to share screen with my PowerPoint slides and see what happens. Let's have a look at that. Here we go. And you should all be able to see the first slide of my talk. There we go. Now, the title of my talk is Pause for Thoughts. It's a bit of a weak pun, but the point behind it is that cells, of course, have to communicate. And the way a lot of cells communicate is indeed by specialized pores in their membranes, which open and close in response to changes in electrical signals. And that really is the core of what we've been working on for the last 20 odd years, and is the main focus of what I'm going to be talking about today. But to give you some context, a typical adult human body contains of the order of 30 trillion cells, which is a very big number. And these cells are all associated together, and they connect together to produce tissues, here, and then the tissues themselves assemble to give organs, and the organs themselves you can think of as part of systems, etc. So that there's this hierarchy of interactions between all of these different components. And a lot of the things that we worry about and work on in biochemistry and cell biology and so on is to try and unpick all of that complexity and try to see whether or not there are, you know, what are the mechanisms by which these interactions work, what are the rules by which they work, and what happens when those rules don't work. And that's why it's particularly important to understand how cells talk to each other. So that really is my point, that the collective emergent behavior of these systems, and I think that is the right way to think about it, as sort of an emergent behavior of these interacting cells, gives you this higher order behavior. And you can't really understand that behavior without thinking about the context of the whole organism. In other words, what is it doing for the whole organism? And the two examples that I work on in particular are nerve communications between nerve cells, particularly in the context of pain perception, and also in the heart, where muscle cells of the heart interact with each other in such a way that they spontaneously generate these fantastic contractions, which of course is the heartbeat. Okay, so what we'd like to understand ultimately is how all of that emergent property comes to be as a result of the interaction of relatively simple molecules. Okay, now that's a big topic, of course, and I've only got 30 minutes, so I'm not going to get too heavily into that bigger philosophical issue, but instead I'll focus on, as I say, these two areas, pain perception and cardiovascular, the heart, and how cells within that system communicate. So in the case of pain, pain has a very important biological function. Okay, we might think that it would be great not to have pain, but actually, of course, it is important. It warns us when damaging things are happening to our bodies, and it motivates us to get out of the way quickly. And the way in which pain is perceived is now beginning to be examined in great detail by a lot of clinicians and molecular biologists. And the reason for that is that pain is a very big problem clinically. Okay, in terms of chronic pain and things like that is a major health issue, which clearly affects quality of life and so on. And there aren't that many good treatments for chronic pain, and so there's a very important area of research to try and find better clinical treatments, and to understand, and for that we need to understand the mechanisms better. So when you prick your finger or you stick a finger on a thorn bush or something, a signal is sent from your hand through ultimately to the brain. But it goes through this little processing center, which looks very peculiar, but this is actually part of what's called the spinal cord. It's actually a specialized subsystem, if you like, within the spinal cord. And in particular, a bunch of neurons called the dorsal root ganglia. And the nerve cells, the neurons, come together to generate an initial processing of that signal. So how does that signal work? Well, the neurons themselves connect together and touch each other, or whether, strictly speaking, they don't really touch each other. They come very close and they form these so-called synapses where there's a little gap between them. So if one neuron fires a signal, it gets to the synapse, there's a release of specialized chemicals called neurotransmitters, which then activate the signal in the next neuron, and so on and so on. So we can think of these neurons firing a particular signal, and whether or not the downstream neuron fires or not depends on the type of neurotransmitter that's released. Okay. So you can see already that there's a sort of potential for generating complexity from relatively simple signaling systems. But how that actual signal works was first worked out actually by two scientists here in Cambridge, Huxley and Hodgkin and Huxley, sorry, in the 1950s and 60s. And how it works is this. the membrane of the cell, or the neuron, is a long, eventually think of it as a long tube, actually. And we can call that the axon. And that tube contains a membrane. And that membrane is impermeable to small molecules like ions and so on. And as a result of that, there's an imbalance between different types of ions. So for example, there's more sodium ions outside the axon than inside, and there's more potassium inside than outside. And because the potassium and the sodium ions can't move across that membrane, because it's blocked, then that builds up a gradient, and that gradient is a gradient of charge. And effectively what we've got here is a little sort of battery, okay, with an excess of positive charges on one face, one side of the membrane, compared to the other. So the net charge, it tends to be more positive on the outside than the inside, but that needs to be understood that there are lots of positive charges inside. It's just that there's relatively fewer inside than outside. That's the point. And as long as this membrane remains intact and can't allow the sodium and the potassium ions to move, then that membrane potential will be maintained. But it turns out that within that membrane, there are specialized proteins, which are our pores, if you like. And these are what are called the voltage-gated sodium and voltage-gated potassium channels. So these are proteins which sit in the membrane, and they can open and close in response to changes in the membrane, in the potential across that membrane. And if they open, they can allow sodium ions into the neuron, in the case of the sodium channel, or they can allow potassium ions out of the axon, out of the cell, via the open potassium channels. Remember that there's more sodium outside than inside, so if the sodium ion channel opens, there'll be a net movement of sodium ions into the cell, and if the potassium channels open, there'll be a net movement of potassium ions from the inside to the outside. And it's that change in the relative ratio of the two ions which gives you this perturbation in the membrane potential. And this is called the action potential. And we can actually measure it by putting microelectrodes into the cell at various points, and we can measure the changing voltage across the membrane. And so that's what this little GIF is supposed to show. This is what the sodium ions, the sodium channels, the blue proteins, and the potassium channels, the little orangey proteins, are doing. And then the sodium ions are the little blue dots, and the potassium ions are the sort of orange dots. So keep an eye on these two components here as they sequentially open and close. So what happens is that as if the membrane potential is perturbed, and that can happen by, for example, the neurotransmitter activating, changing the membrane potential, then what will happen is that the sodium channel will open, and that will allow the sodium ions in. So that's what's happening here. And that will then change the local potential in that local area. But within a few milliseconds, that's a few thousandths of a second, that sodium channel switches itself off. It goes into what's called the inactive state. So it can't work anymore. And at that point, the potassium channels open, and now the potassium ions start to leak out. And the result of that is that the net charge rebalances. But in doing so, that then perturbs the membrane potential downstream of that. And so what happens is that you get this wave of depolarization, as it's called, as it goes down the axon. And that essentially is how nerves talk to each other. That's how the action potential works. And that is the central process that underpins, well, frankly, underpins everything that that neurons do. That Bill Cattrall, who's one of the great pioneers in this field, is fond of saying that everything that you think, or your thoughts, your feelings, your emotions, everything depends on the opening and closing of these sodium and potassium channels. It's a pretty awesome thought if you put it like that. So the other major point, and this is where I think it gets particularly exciting, is that for a long time we've been stuck in a relatively crude idea of how this works, with little cartoons of squares and circles opening and closing. That's only takes you so far. What scientists would really like to do is to look at atomic detail or new molecular detail, because that gives us the clues as to how these channels actually work in practice. It turns out that this is a very difficult problem, and part of the reason is that these proteins are very large and they sit in membranes, and it turns out it's very, very difficult to get structures for these types of proteins. But recently, in the last few years, and again, I have to say, researchers here at Cambridge University largely have developed new techniques, and a particular technique called cryo-electron microscopy. There's one of the pioneers of this field, Richard Henderson, works at the LMB in Cambridge, and he won the Nobel Prize a couple of years ago for this work on cryo-EM. And I believe actually Dima Shagazi is going to talk a little bit about cryo-EM too as well in one of his talks. So anyway, now that this is a fantastic time, because now finally all of these sodium channels and ion channels and potassium channels, all the rest of them, they're all tumbling out. These structures are being published every couple of months, and we can start to see in amazing detail how they work. Now, that's a massive presentation in its own right, and I don't have time to go into all of that, but suffice it to say, just to give you a taste of what we're looking at, this is what the sodium channel now looks like. This is a rendering of it, obviously a diagrammatic presentation in which different parts of the molecule have been selectively colored so that you can make you can understand it a bit better. So this is the so-called top view. That is to say, if you were sitting in the outside media and you're looking down at the cell from the top, this is what the sodium channel would look like. If you're looking from the side and there's the membrane and our little dots, that's what it would look like. Now, the key point is that this hole here, that's the pore. That is literally the pore. And that is just wide enough to allow a sodium ion to move through it when the channel opens. And the opening of the channel turns out to be involved in subtle rearrangements of these molecules here, these so-called alpha helices. It's a bit like an iris, in fact, in a camera. It just opens and twists open and closes in response to these changes in the membrane potential. And that subtle change is enough to open a pore and allow sodium ions to move into the cell. And this is the potassium channel. Now, it turns out that the potassium channel is a distinct protein. It's got lots of differences, but if you look at it, it clearly is very similar. It's got this same four-lobed structure. And again, you've got this pore in the center, and that again is where the potassium ion moves. The selectivity, the reason why the sodium channel can only allow sodium ions through and potassium channels can only allow potassium ions through is because of very subtle differences in the structure of that pore. It's very, very subtle, but it's just enough to give you fantastic selectivity. Nature has clearly hit on this very a neat idea of just tweaking a few residues in that pore to give you different specificities, even though the overall structure is very, very similar. Now, the other reason why these new structures is particularly interesting and important, and the connection with pain, is that there is a particular type of sodium channel. It's called NaV 1.7. And this particular type of sodium channel appears to be specialized for pain perception. I should say, by the way, that there's about 10 different types of sodium channels, and they're expressed in different parts of the body. Some are in the brain, some are in the muscles, and so on. This particular one, NaV 1.7, is found largely in those pain-sensing neurons that I showed earlier in the dorsal root ganglia. And the reason why we're getting very excited about this channel is because there's a very, very rare genetic condition in which people literally cannot feel pain, okay? The problem these people face, as I hinted at earlier, is that because they can't feel pain, they don't realize they're doing damaging things to their body. So they put a hand on a hot plate and don't realize that they've burnt their skin and things like that. And so it turns out it's a very, very difficult condition to live with. But the great breakthrough that studying these rare patients has done is to give us a new insight into the importance of this NAV 1.7, because these patients lack a functioning NAV 1.7. And this is also, so that tells you that NAV 1.7 is absolutely central to pain perception. But it's also telling you something more and more interesting, because these patients are otherwise normal. They have normal IQ, they develop normally and all the rest of it. It's just that they can't feel pain. So that tells you that this NAV 1.7 must be highly specialized for pain perception, because if it was doing other things as well, you'd have additional developmental problems and so on, but you don't. And that means that if we could get a drug that could target NAV 1.7, we might have a really good, very selective pain analgesic, something that could target a pain receptor and not have many side effects. And so that's actually a major research project now among lots and lots of people. And indeed, actually, we now realize that some very well-established local anesthetics, for example, work by blocking these pain sensing sodium channels, lidocaine, which you go to the dentist. Turns out to be a sodium channel blocker. More interesting, more importantly, perhaps, there's already a very large number of sodium channel inhibitors out there as a result of, of natural the molecules produced by animals and plants and so on for their own purposes. And the most notorious of which is this compound called Tetrodotoxin. So this is a small molecule that's made by the pufferfish. Okay. Now, it turns out that the toxin gland from the pufferfish is, or rather the muscles surrounding the toxin gland, turns out to be a very highly prized sushi delicacy. The problem, of course, is that tetrodotoxin is one of the most powerful poisons known. In fact, in terms of, molecule for molecule, it is, I think, literally the most toxic compound known. And the reason it does so is that it binds to many sodium channels. It binds very, very tightly, and it basically stops them from working, which means that you won't be able to breathe, you won't be able to see, all the rest of it. And indeed, tetrodotoxin poisoning and by tetrodotoxin is a very nasty way to die. If you want to try fugu, which you can do if you go to Japan, make sure you go to the right restaurant. To be a fugu chef, you have to train for years. It's a skill that's very tightly regulated, not surprisingly. My advice to you is don't try this at home, okay? The reason, on the other hand, tetrodotoxin turns out to be a very useful reagent, not so much for therapy, but for experimental work. Because by blocking sodium channels in experimental systems, you can often unpick how sodium channels are working. And tetrodotoxin, or TTX, is one of the compounds that's often used for this purpose. More promising as potential therapies, there's a whole set of very interesting molecules that are found in animal venoms. In particular, the cone snails and spiders. This is the toxin from the cone snail called contraction, and this is a toxin called Huon toxin from the tarantula spider. And these also target sodium channels. But it's important, I think, to realize that, they didn't evolve these toxins for our benefit. They evolved them because they were useful to the animal and the snail and the spider. And often these toxins are actually primarily targeted against the muscle sodium channels. In other words, they're designed, as it were, to induce paralysis in the victim that the cone snail or the spider will bite. But it turns out that some of these toxins, and the Huon toxin is a particularly useful one in this respect, do also target the NAV 1.7. So the plan might be to see, well, perhaps what we can do is we can change the sequence of that Huon toxin tweak it by molecular techniques. A technique called site-directed mutagenesis will allow you to selectively change the sequence and therefore the structure. And there's a lot of interest now in biotechnologists and pharmaceutical companies to start playing around with some of these toxin molecules and see if we can make them more specific so that they will only target the pain sensing 1.7 and not any of the other sodium channels. And for that to work, you do need a very detailed structure of the channels, which is why these atomic resolution structures that are now tumbling out from all the structure labs is so important and has sort of amplified that area of research. One final point, though, as well as venom toxins which inhibit channels, there are some remarkable examples of venoms toxins which do the opposite effect. So this is a toxin called Potassium, and it's produced by a South American ant called the bullet ant. And what it does is that it binds to NAV 1.7 and it does the opposite effect. It locks the channel in the on position. So the effect of that is to induce what's said to be an excruciating pain. It's been likened to the pain from a gunshot wound. And indeed, that's why it's called the bullet ant. There is the various Amazonian tribes have a sort of initiation ceremony where they dare each other to put their hand into a glove full of these bullet ants. This is taken from an Australian comedy show called Hamish and Andy's Gap Year, which you may or may not have seen, but the comedian, this comedian, decided it'd be a good idea to experience an Amazon's tribe's coming of age ritual. And so live on television, he did that. I'm not going to tell you what happened, but if you're interested, it's on YouTube, what could possibly go wrong. I also work on the muscle sodium channels and the muscle ion channels, in particular, the heart. Now, the heart is an amazing structure. It beats literally, actually from before you were born, well, literally till the last minute of your life, right? And it continuously beats like that all the time, and without tiring or without changing its behavior. And that, of course, is absolutely critical to keep your blood pumping. So understanding how the heart works and the heartbeat works is another major area of research in cardiobiology and cardiovascular biology. And the muscles of the heart are absolutely amazing. So you can take them, if you look at the muscles of the heart under the microscope, you can see these cells, the so-called myotubes, which are really just cells connected together in series to form connections which are electrically connected. And then the tubes themselves are connected in parallel to form these little bundles. And it's the combination of that that allows the muscles to coordinately contract and expand, okay, to generate the heartbeat. And you can even take some of these cells from the heart and you can grow them in tissue culture, or you can just put them in a tissue culture and they will spontaneously beat. You can also look at them, and so this is actually not, strictly speaking, this isn't a real live cardiomyocyte. This is what's called an induced pluripotent stem cell. So this is a special type of cell which has been induced to turn itself into a heart cell or a cardiomyocyte. And again, one of the features, one of the reasons why you know that this has worked is when you start to see it spontaneously beat in the microscope. So to understand how that works, we need to think about, again, how the cells are talking to each other within that muscle bundle. And it turns out that this time you still need the sodium channel and you still need the potassium channel. So you have action potentials as usual. But now you also have a third ion channel called the calcium channel. And the calcium channel is sitting there as well. And it opens in response to the change in the opening and closing of the potassium channels. And the calcium channel opens to allow calcium in, and that's the trigger that actually activates the contraction. Well, again, now the structures of the calcium channel is coming out. And again, it turns out it's again the same sort of structure as in the sodium channel and the potassium channel. In other words, all of these channels essentially are evolved from the same common ancestor. They're what we call homologous. They evolved by a process called gene duplication and divergence many, many millions of years ago, and to become specialized. And again, there's the pore there in the calcium channel. And again, sequences, little regions around that pore are different, again, between the calcium channel and the potassium channel and the sodium channel. And those differences are enough to give you these differences in specificity. So what will happen then if you have your sodium channel and potassium channel and your calcium channel now in the same membrane, and again, we can start off with what's called the resting state, where all of them are closed. If the sodium channel opens, then that begins the depolarization, that begins the action potential. And again, this time what happens is that activates the opening of the calcium channel. And the calcium channel then lets calcium in, and it's the calcium which is activating all of the contraction, etc. And eventually, though, the potassium channels open, and that brings back down the action potential and switches the system off. So this is an experiment in which, again, these cardiomyocyte cells, as they're called, have been grown in tissue culture, and we've induced into it a special dye that responds to calcium. In other words, we can see the changes in calcium as these cells beat, and you notice how they're beating spontaneously. And notice how these bursts of calcium appearing in the cell and disappearing in coordination with that beating. And that's because the action potential is driving the opening of the calcium channels, stimulating the release of calcium, and that then activates the muscle contraction. So the sodium channel, potassium channel, calcium channel are all acting together to generate the heartbeat. It turns out then that a lot of the problems that you run into in cardiobiology and cardiopathology can be traced to some of these ion channels. There is quite a number of inherited conditions which cause inherited cardiopathologies. The most famous example is called Brugada syndrome, after the Italian scientist who first discovered it. There's another one called long QT syndrome, and so on. Sick sinus syndrome is another one. Now, these are all inherited diseases that are relatively rare, like the pain insensitive patients. But nevertheless, they're very instructive because they give insights into how these channels work in practice. And the particular patterns that you get with these mutations and these diseases vary between the types of channels that are affected and the position on the channels that are mutated. And that gives us very important clues as to how these channels work, and also gives us new insights into how potentially we could generate new therapies and new drug targets to correct some of these underlying deficiencies. And just to give you a very simple example of this sort of thing, this is actually work from our group. This is the heart-specific sodium channel. So there it is in the membrane, and there's that structure that I showed earlier. Now, I don't want to get too into this because it looks very complicated, but basically what these residues are, these are specialized amino acid side chains which stick to each other and connect the little loops in the sodium channel. And it's important that they do normally stick together because that makes sure that the sodium channel folds in the correct way and functions in the correct way. Now, it's been known for about 20 years that there are some of these Brugada mutations which occur in this local region of the channel. And nobody really could understand why those mutations were causing this condition. But now the structure of the channel has come out, and we can see where those residues are. And these are all independent mutations. And lo and behold, it turns out that they all form this nice little cluster, and they can stick together to form these connecting interactions like sort of Velcro, if you like, molecular Velcro. And there's a particular type of Brugada syndrome where this particular residue, an arginine R881, is mutated. And there's another type of Brugada syndrome where this residue, which is a so-called aspartic acid, is mutated. And these mutations have been known for a long time, but it's only now literally in the last 12 months, when we start to look at the structures and we can finally understand why they're causing the mutations. Because by breaking that little patch of interaction, you destabilize the whole of this channel here. And so the channel can no longer undergo the conformational changes that it needs to open and close in the way that I've explained. So that's one of the reasons why these structures are really interesting and they're interesting in their own right, but they're also very important clinically because it immediately suggests completely new avenues for therapeutic interaction. So the take-home messages then are the nerves and the muscle cells talk to each other using electrochemical signals. We call that the action potential. And the sodium, potassium, and the calcium channels are all playing important roles in this process. And any one of them can break down and generate various types of pathology. We know that there are lots of drugs that can target these channels, and some of those drugs are based on animal toxins which naturally target the channel. And some of these toxins can then be retooled, as it were, or remodified to be more specific for a particular ion channel that we want to target. And so that's an incredibly useful, very powerful technique for generating new drugs. And the final take-home message is, don't make fugu at home and never, ever put your hand in a glove full of bullet ants. Okay, so I just want to say thanks to everybody who's been involved in this work. Some of the work I've presented actually is literally from my group that we're doing at the moment. So Samantha Salvage has been working on the heart-specific sodium channel. Samir Hamir has been working on making so-called single-chain antibodies to these channels. Zaki Habib is a graduate student working again on the heart-specific sodium channel. Hengri Liu is a graduate student, again working on the heart and pain-sensing sodium channel. And Manas Kohli has been using computers to model how the sodium channel works. And then our long-term collaborator, Professor Chris Wanger from physiology, who has been a long-standing expert on the heart-specific sodium channel, has been working with us now for some time. Just for completeness, this work has also been funded by the British Heart Foundation, which is why I gave a little sign out to one of their websites. Wellcome Trust, Newton Trust, and actually a very interesting and a fruitful collaboration with AstraZeneca and a company called Medimmune. There you go. Thanks, Rhys.
Great, thank you very much, Tony. That was a really interesting talk, and I absolutely love your graphics and animations. It made it very easy to follow and understand. So that was fantastic. Thank you very much. So we're now going to have our live question and answer session. So if you have any questions, please just post these in the live chat on YouTube, and then we'll try and answer as many of these for you as we can. And there is a 20-second delay, though, between what we say on Zoom and you hearing us on YouTube. So I'm going to kick off the question and answer session. And so I guess my first question, Tony, is why sodium and potassium? Is there something special about these ions that make them particularly useful for the cell? Or
is an interesting question, and I suspect it's to do with their ubiquity. Okay, sodium, of course, is present in essentially all liquids, seawater, and so on, and potassium likewise. The other thing about sodium and potassium ions is that their sizes are different, but also what's different is the number of water molecules that surround them, the so-called hydration shell. So if you're a protein that's trying to distinguish between a sodium ion and a potassium ion, it's not as difficult as it might look initially because the sizes of the hydration shell are different. Okay, so you can imagine a basic channel, and we know that all of these channels essentially function in the same way. That is to say, they have this what we call fourfold pseudosymmetry with a single hole in the middle. and that hole opens and closes, and that pore has to be wide enough to allow the right ion channel through and stop all the other channels going through. Yeah, so the sodium and the potassium ions are different enough in terms of, first of all, the density of their charge, and also the hydration shell of the water around it, that it turns out it's a relatively simple way, simple to select against them. Calcium is another more interesting in some ways, because the cell goes to a lot of trouble to keep this amount of calcium inside the cell very low normally. That means that there's a very, very steep gradient of calcium outside the cell and also inside in specialized organelles which package the calcium. And so when the calcium channel opens, there's a really very rapid movement, or a very large, if you like, potential to push the calcium's ions into the cell. So the calcium level goes inside a cell from very, very, very low to spike very high and then come back down again as it's pulled out. And that again is something which means that you can, once you have that system established where you have a very, very large difference in the concentration across the membrane, it means that it can be a very useful signaling molecule.
I see. And actually, you mentioned the channels in your answer there. I have to say, from your slides, it was impressive just how similar they looked. Particularly the top-down view; they're almost identical.
They are, and in fact, you can. I don't know. Can I go back to the?
Yeah, of course.
Right. Okay. Let's see if I can go back.
You just need to share your screen again.
Oh, okay. Here we go. Yeah, here we go. Yes, so indeed, when you look at them, superficially, they look very, very similar. In fact, you can actually take that and superimpose it. The differences are largely concentrated in the pore region. There are differences in these regions too. These are what are called the voltage sensors. So this is the part of the molecule that actually physically senses the changes in the membrane potential. And it's the movements in those regions which kickstart that whole process, which opens up the iris, if you like. And so there are differences between the ion channels in those regions and in those regions as well. But there are also remarkable similarities, as you say. The broad structure is essentially the same. And again, that's overwhelming evidence that all of these ion channels basically evolved from a single common ancestor.
So do we know which one came first then? And perhaps, and also, I guess, what was that first channel used for?
Well, I don't know is the answer, but there is a lot of work now done by evolutionary biologists who understand how to analyze sequences properly rather amateurishly like we biochemists do, you know, and you can actually get some insights into the evolutionary origin of these things. It seems to be the ion channels, sodium, potassium, and calcium do seem to be a eukaryotic invention, although having said that, there are bacterial channels as well, which look very similar, and probably so. In other words, the deep origin of these channels goes back even further between the eukaryotic and prokaryotic split. But those bacterial channels are a bit odd because it's not clear that they are actually ion channels per se, but they're clearly structurally related. So this is a protein which goes long way back.
I guess that makes sense because action potential is going to be a very critical function to higher organisms.
Exactly. I mean, any cell that has a membrane is likely to generate an action potential or a difference in charge across the grade of the membrane, just because, you know, the movement of the ions will be restricted. And so a membrane potential is something that can emerge relatively easily. But once it's emerged, it's a very useful source of potential energy, of course, because by changing the membrane potential and discharging it when physiologically necessary, you've got a terrific way of generating a signal.
Thank you. I was going to say, you've stopped showing your screen, but one of your first slides, you mentioned using microelectrodes to measure the action potential. How do you get the microelectrode into the cell?
Well, that was clearly a sort of diagrammatic process. Actually, Hodgkin and Huxley, for some reason, I don't really be able to say that anymore. But anyway, what they worked on was the giant squid axon. Okay, now the giant squid axon, as the name implies, is very big. And so you can actually put microelectrodes inside that. Yeah. But in most situations, you would use what are called patch clamp electrodes. So you put an electrode on the membrane and suck out the membrane. And you can also inject charge into the cell so that you can manipulate the membrane potential that way. So that's how one would normally do these types of experiments in a modern setting.
Yeah. Okay, I was picturing sort of something like trying to thread a needle, but on a very microscopic level. There's an interesting question here on YouTube. So can biochemistry be used to find the causes and new treatments for inherited diseases?
Well, absolutely, and I gave a few examples there. The discovery of these patients with NAV1.7 mutations is a good example of that sort of approach. These patients are very rare, okay, but because they are sort of natural experiments, if you like, if you study them and you find out what the mutation, where the mutation is and what it affects, it gives you important insight, general insights into the whole field. So if you're starting with something like pain, how does pain work? That is a huge, big problem. And it's often, when you're dealing with these complicated biological problems, it's often difficult to know where to start. And one of the great things about these natural human mutations, which crop up all the time, is that they give you a toe in the door. They give you a clue as to how the system works. And oftentimes the clue is initially quite surprising. So when researchers started to look at these patients who literally can't feel pain, and they started to take DNA samples and they started to compare their sequences between members of the same family who could feel pain and so on, they were able to identify a consistent mutation which tracked with the insensitivity to pain. And that turned out to be a mutation which affected this NAV1.7, this sodium channel. And that was the first insight into what's going wrong in this particular condition. The other point, of course, is that once you've identified this protein, you can then have a look and see, well, where is it expressed? Oh, look, it's in the pain-sensing neurons, right? And all of that sort of stuff. And then you say, oh, wait, look, some of these drugs which you've been using for anesthetics and also are known to affect pain sensation, they also bind to this channel. Right? So it's like a jigsaw puzzle. All the bits and pieces suddenly clunk. You've got the whole sky and a bit of the mountain suddenly.
And is that the same then, presumably, for NAV 1.5? There are drugs against that?
Yeah,
that
absolutely.
Heart disease.
Yeah. And indeed, the same thing applies there. These natural mutations, these Brugada patients and long QT patients, there are hundreds, and it wouldn't surprise me if there are 1,000 individual mutations in NAV 1.5 which cause these conditions. And what is interesting actually is that the different types of diseases, the Brugada versus the LQT, the mutations occur in different parts of the sodium channel molecule. They're not randomly distributed. They're different parts appear to be mutated, and that will give you LQT, and other parts of the molecule are mutated, and that will give you Brugada syndrome. Okay, and that's got to be an important clue. That's telling you something very interesting and important about how these channels work. Right? And so that's one of the reasons why a lot of work is done on these channelopathies, as they're called, but also because now that we have these structures, it means that we can start to think about the sodium channel as a potential therapeutic target.
Thank you very much. Latika, do you have a question that you want to ask? Or?
No, I think I just want to say it was a really, really good talk, and thank you for giving one. I remember tuning into your talk a few years back, and again, it was just one of those things that I found really informative and entertaining, especially with the bullet ants. I mean,
I think it's still on YouTube actually, but
yeah. I highly recommend to anyone who's watching right now to try and find that video of the guy putting his hand into the glove, because it is just ludicrous what he's doing.
Someone should post it in the comments on the video to share it with other viewers. I had one other question actually, Tony. So you mentioned that you use the tetrodotoxin in research. Presumably that must be extremely tightly regulated then.
Yes.
If food is tightly regulated.
Yes,
so
yes, it's locked away in the poison cupboard.
Yeah.
Yeah.
And one final question, I guess, because the neurons and the heart both have sodium channels. Does the heart feel pain?
That's a good question, actually. I don't think it does, actually. That is to say, if you, I guess if you're doing an operation and so on, you wouldn't feel pain in the heart or so on. I guess you'd feel pain as you went through the skin and everything, but of course you're under anesthetic.
Interesting.
I mean, to be honest, it's a bit more complicated than that. So, for example, the NaV1.5, which we think of as in the heart, it turns out it is actually also expressed in the brain and the central nervous system. It's in specialized cells. We're still not quite sure why that is. Oh. And amazingly, the NaV1.7, the so-called pain-sensing sodium channel, is expressed in the beta cells of the pancreas, of the islets of Langerhans, which are involved in insulin secretion. Okay. Now, why that should be is a big mystery, but it does look as if there is increasing evidence that that is doing something important in the process of insulin release and in response to changes in blood glucose. So, we shouldn't perhaps pigeonhole these channels as saying, this is the pain, this is only involved with pain perception, or this is only involved in heartbeat, and so on. Nature doesn't care about that. Nature will use whatever is useful, but the more and more we look at these things, we find additional little interesting, quirky things about them and additional functions and so on.
Perhaps that's something for a future Cambridge Festival.
Yeah, well, that's a PhD project for somebody, right?
Yeah.
If you're interested, folks, drop me a line.
The contact details are on our website. I think it's time to wrap up this session, actually, because we're a little bit over. So I just want to say thank you very much again, Tony, for a fantastic and very visual talk. That was great. Thank you. Thank you also, Latika, for helping out behind the scenes again.
No problem.
And thank you very much to all of you at home or in your gardens for watching this talk. So hopefully we'll see you again later this afternoon or later this week for the rest of our Cambridge Festival talks. So stay safe.